Right, thank you very much for the introduction. I hope you can hear me well, and I hope you can now see my screen. It worked well when we tried the sharing earlier. So what I would like to talk about today is a problem we have been working on for a while. So imagine that you could look at a sequence or two sequences that encode the same protein in any organism of your choice. And you could tell which of those sequences is more efficient. So which makes more protein, or possibly which makes better protein, which sometimes is a different problem from making more protein. So what I'm trying to tell you today is, or what I aim to tell you is that basically we can do that in many contexts. And I will try and explain how we go about doing that. So one of the things we focus on when we try and interpret the efficiency of genes is the movement of ribosomes. And so here, this is a very simple animation, which is probably the output of about, well, 16 years of work, which is realistic in the sense that it depicts a gene. The small dots are genes, the small dots represent a codon each, and the large dot wandering along it is a ribosome that decodes the gene. And what's realistic about this animation is that the step size and the relative width of the ribosome is realistic, and the step time is realistic. So the time it takes the ribosome to decode each codon is realistic as far as we know and according to our models at the moment. And you will see that right about in this region here, there's a slow stretch where the ribosome moves very slowly, whereas on the other codon it moves fast. And this is also realistic. So I've built a sequence here which contains some typically fast-moving codons. So this stretch would be some of the fastest codons you see in a cell, but then this stretch would be some of the slowest codons you can actually see in the genetic code. So one interesting problem about this is that if RNAs are not usually translated by a single ribosome, instead, when one ribosome starts moving along, a second one can initiate and can also translate the same gene. And if you have combinations of fast and slow regions, then you get things like this. You get collisions between ribosomes, and that's generally bad news. Okay, it's bad news because the ribosomes have sensors that allow them to detect when they touch another ribosome. And that is usually interpreted as a problem because it may be the result of slow codons such as here, but it may also be the result of a chemically damaged RNA or a chemically damaged ribosome. And usually two ribosomes contacting each other leads to the destruction of the RNA and the ribosomes in question. It's a safety mechanism in the cell. And so when you think about RNAs that are optimal for function, if you want to express a recombinant protein, for example, you want to avoid this kind of problem and you want to make the flow of the ribosomes along the gene usually much better than this. So you want to avoid collisions. So I'll take a step back now. In general, in eukaryotes, which we are mostly interested in when you want to translate an RNA, there's quite a complicated process that happens, which starts by preparing the small ribosomal subunit to initiate at the cap structure of the RNA. The subunit associates with some translation initiation factors, eukaryotic initiation factors, or EIFs, recognizes the cap structure of the RNA. Then there is a process called scanning, where the small ribosomal subunit moves along the untranslated region until it finds the start codon, and the large ribosomal subunit joins. Then you get synthesis of the protein, and on the stop codon, you get dissociation of the complex and release of the newly made protein. So in a way, there's two sub-processes here. The initiation process at the beginning, which makes contact between the ribosome and the RNA, and then the protein synthesis process itself, which makes the protein you're interested in. If you think about regulation in these processes, there's basically two regimes of regulation that can happen, and they depend on what the frequency is with which ribosomal subunits make contact with the RNA relative to the speed with which ribosomes move along the RNA. And so you can have one regime here. Sorry, I'll just switch my laser pointer on. You can have one regime here where you have relatively slow initiation and relatively fast movement along the RNA. And in this regime, if you want to make more protein, you need to increase the initiation rate. If you increase the elongation rate here, nothing happens to the amount of protein you make because this process is not limiting. And the other regime that we're thinking about often is this. You have very fast initiation, so the processes which establish contact between the small ribosomal subunit and the RNA are very efficient, and movement is relatively slow relative to this process. Where that happens, the ribosomal movement along the RNA determines how often you can initiate, because this process cannot be faster than the movement of the ribosomes away from the start codon. And so in this regime, if you want to make more protein, you need to accelerate the ribosomes. With physiological rate constants for initiation and elongation, we can show that you can switch between these two regimes. And in a cell, there are typically some genes which operate in this regulatory regime and some genes which operate in this regulatory regime. We have quantified this using data that were published. So these are not our data, but we reinterpreted them. And the data were generated using a technique called ribosome footprinting. And in particular, this data set is for small subunit footprinting. So the way the data are being generated is that in the cell, small ribosomal subunits are cross-linked to RNAs, and they're isolated from the cell. And the RNA is then digested away using nucleases, but where there is contact between the small subunit and the RNA is protected from digestion. And so when the protected subunits are sequenced, essentially during initiation, ribosomes move quite fast along the untranslated region, but then stall over the start codon while they're waiting for the large subunit to join. And as a result of that, essentially you see a dominance of sequences that are around the start codon, because that's where the small ribosomal subunits protect the RNA most. And so when you do this and you map, this is a pile-up graph of different sequences recovered from the seq experiment, you map where the sequences are being recovered. This is all around the start codon for most genes, and all of this is in Baker's yeast. But for a small number of genes, you can actually see that there is this peak again where the start codon is, but there is a second peak ahead of the start codon. And our interpretation of this peak is that this is where there is small ribosomal subunits queuing, because this ribosomal subunit cannot move away fast enough because there is other ribosomes queuing ahead of it. And so if you think, and so the ratio of these two genes we see in Baker's yeast is about 5:1, roughly. So essentially we think that this peak represents genes which are in this regulatory regime, because here there is a blockage here and then small subunits which initiate queue and need to wait. And so in vivo, in natural RNAs in Baker's yeast, we think that about 1/5 of RNAs are in this regulatory regime and about 4/5 are in this regime. Interestingly, in biotech constructs, so whenever you want to do recombinant protein expression, almost all constructs are in this regime, and that's simply because the constructs used to generate, so the regions for translation initiation, for example, used in recombinant constructs are very, very efficient. And so these processes are very fast. And so you need to optimize your coding sequence. You need to codon optimize your coding sequence if you want to get the best expression levels out of your gene. So how do we, so in this situation, which applies to many recombinant protein expressing constructs, clearly it would be useful to understand how rapidly ribosomes move along your sequence and to make sequences where ribosomes move particularly fast. And that's what we're trying to do, and we do this with computational models. And so our aim is to model the movement of ribosomes along the mRNA. In order to understand this movement, we need to understand the sampling process by which the ribosome selects tRNAs. So this is supposed to be a representation of a tRNA that come in and out of the ribosome, and where the ribosome tries to match the anticodon region of the tRNA with the codon that sits in the ribosomal A site. So this is the sampling process. Ribosomes don't have any eyes, so they can't select the correct tRNA that needs to come into the A site here to match the codon. And so what happens is a random tRNA enters the A site. The ribosome evaluates whether the anticodon matches the codon. And if it doesn't match, then that tRNA is rejected. If that anticodon and codon match, then the tRNA is accepted. This peptide is transferred onto the incoming amino acid, and the whole ribosome shifts forward by one step, and the sampling process is repeated again. So if we want to understand movement, we need to understand the sampling process. And the sampling process is a result of two things. One is the biochemistry with which the ribosome evaluates tRNAs when they come into its A site. And we understand that biochemistry quite well because very good biochemists have studied this in detail over the years, and we know all these rate constants. But another aspect we need to understand is the base pairing relationship between the tRNA anticodon, so this bit of the tRNA, and the mRNA codon, so the part of the mRNA that sits in the A site. Different tRNAs have different rate constants in this scheme depending on how they base pair with the codon. And as a rule, the stronger the base pairing is between the anticodon and the codon, the faster the forward rates in the scheme get, and the slower the reverse rates get. Whereas if anticodons do not base pair with the codon, then we have slow forward rates and fast reverse rates, which means that the tRNA tends to enter the ribosome and go out again. So I said we use computers to model the decoding process to interpret all of this information, and that's actually not a new thing to do. The first computational model of ribosome movement was published in 1969, the year I was born actually, which I've always thought of as a good starting point for me to start getting into this discipline. But the computer that was used at the time is actually this, and this man here is not sitting in front of the computer, he's sitting in the computer because the entire room is the computer, and this is your hard disk, and this is your processor, and this is your RAM essentially, and the whole thing had a whopping working memory of 64 kilobytes. So what people were modeling at the time was a very, very simple version of ribosome movement, but they still used the same principles that we're using today. We've just made the schemes, the reaction schemes, more complicated. And just for fun, I was looking for other things that have as little computer memory as this huge computer had at the time. And almost the only thing we can find these days is that some toasters use microchips to tell how brown the bread is that you're trying to toast. And they may have, if they have small chips, then they have a similar working memory. But your typical computers will have about 1 million times more memory. So I'm always very impressed by what people did with almost no computational power at all many years ago. So we now have a lot of computational power. And so when we do our computational models of the decoding process, we can do quite complicated things. And essentially how we get to our end goal of modeling ribosome dynamics is that we need to define quite a number of things. We need to establish what tRNAs are present in the organism for which we want to model ribosome movement. We need to establish how the tRNAs pair with the different codons. And so in Baker's yeast, there are about 45 different tRNAs, which decodes the 61 codons. And so you need to define 61 times 45 anticodon-codon relationships. So this is quite a large data set. You need to know how abundant the different tRNAs are in the cellular mixture, because that determines how likely they are to enter the ribosome before other tRNAs do. You need to know the reaction rate constants, which we take from the literature, that tell you how different tRNAs react in the ribosome. And with this information, you can predict codon decoding times for individual codons. And if you then also have sequence information for an RNA sequence, say, you can predict how these codon decoding times give rise to the ribosome dynamics. And that's our ultimate goal, essentially. So one of the problems we encounter in doing this is that in tRNAs, nucleotides are very heavily modified. So just to illustrate this, I've shown you here some of the uridine modifications that exist. Most of these exist, but not all of this exist in bagaces. Some of these are in evolution domain specific. So here's a plain vanilla uridine, the uridine that you find in any RNA. And you can see in all of these other uridine variants, the modifications have been highlighted, and they may include methylations to the ribose, modifications in the base, which can be exchange of atoms, desaturation of bonds, taking the entire base and turning it by a third of a turn, which is an interesting modification, adding various extensions to the base. The reason why tRNAs contain so many modifications is because these modifications help the ribosome distinguish correct and incorrect base pairing patterns between the codon and anticodon. So I've illustrated the effect of one tRNA modification in detail here. So this is again a uridine nucleoside this is an adenine base, and so you can see the standard Watson-Crick base pair forming here, the standard hydrogen bonding pattern for a Watson-Crick base pair. In DNA, because in DNA the orientation of the opposing base pairs is strictly controlled, this is the only base pair that can form. But in RNA, where you don't have strict alpha helices, you can have many different types of helices, or now, of course, the RNAs can be often single-stranded. In RNAs, opposing base pairs can take on different configurations, and you can, for example, have quite efficient base pairing between uridine and guanine. So this base pairing doesn't happen via the normal Watson-Crick phase, such as the UA base pair you're familiar with, but in the UG base pair, there's different phase of the base being used. So one of the modifications that exists to uridine is this 2-thiouridine modification. This oxygen is replaced by a sulfur group, sulfur atom, and this modified uridine can base pair with adenine in completely the normal way, but sulfur is a much worse hydrogen bond acceptor than oxygen is, and so when this modified uridine tries to base pair with a G, one of the hydrogen bonds doesn't form, and so this base pairing is now much weaker than for the normal uridine, and so this modification has selectively destabilized a wobble interaction. So these non-standard base pairing interactions are also referred to as wobble interactions. And almost all of the modifications I showed you in the previous slide, when they occur in the anticodon region, are there in order to control, either weaken or sometimes strengthen specific wobble interactions. tRNAs are very heavily modified, not just in the anticodon region, and modifications elsewhere have to do with the tRNA stability, but also with recognition by tRNA synthetases, so the enzymes that load the correct tRNA with the correct amino acid. So in summary, for the tRNA modifications, we need to understand how these weird modifications affect base pairing if we want to correctly interpret the base pairing patterns between anticodons and codons, and that's at the moment still one of the limiting things in our computational models. But also these tRNA modifications really interfere badly with reverse transcription, and most RNA quantification approaches these days use reverse transcription to first make a cDNA and then quantify the cDNA by sequencing, for example. And because reverse transcription doesn't work well with modified RNAs, quantifying tRNAs is much more difficult than quantifying mRNAs, say, which aren't so badly modified. I told you that we need to understand tRNA levels to do our models correctly. So again, we have a problem here arising from the modifications. And so what I've shown you here, so people have nevertheless tried to quantify tRNAs, and they have developed different approaches for overcoming the modification problem. And here I'm comparing some of these approaches. So NC-seq is just not caring about the approaches. You just sequence your modified tRNAs, assuming they're normal. There are then different variants of removing the tRNA, or nanopore sequencing can sequence the RNA itself without reverse transcribing it. And so it gets around the problem of the reverse transcription not working so well. In Baker's yeast, we can also just count gene copies for each tRNA. So in Baker's yeast, some tRNAs are encoded by a single gene, but the most abundant tRNAs are encoded by 16 identical isogenes. And in yeast, there appears to be little transcriptional regulation on polymerase 3, so on tRNA transcription, which means that a tRNA encoded by 16 genes should be 16 times as abundant as a tRNA encoded by one gene, and we can use that to approximate tRNA levels. So if we think of all of these as different tRNA abundance data sets and we compare them, you can see that often they yield quite different results. They don't correlate well. Almost the only exception is that this approach called MIM-seq correlates quite well with the counting of gene copies. So these data sets give quite a good correlation. Everything else has real problems, and when you try and compare the tRNA abundances. So we can put all of these tRNA abundances into our computational models, and we get out a data set something like this, where for each codon, so AAA, for example, encoding a lysine, we predict it's encoded in about a 20th of a second, 52 milliseconds. This is a specific data set published on the basis of the gene counting, the tRNA gene counting a while ago, but we can do the same for all the other tRNAs, and we can then compare how good our reactions are. And in this experiment, we have compared our predictions of decoding times against some experimentally produced estimates of codon decoding times. And these experimental estimates were reduced using a technique called ribosome footprinting. And here we have basically used an approach called dimension reduction to take these data sets that predict codon decoding times and ask how similar are they. And in this projection, 2 points which are close together are very similar to each other, and points that are far apart are very dissimilar. And so these are the 5 experimental data sets for codon decoding times. And one of the things you can see is that there are 3 data sets that are quite similar to each other, 2 data sets which are less similar to these codon decoding times. And if we then predict codon decoding times using the different tRNA data sets, we get these points here, and some of them, again, they're all showing different results. Some of them are closer to the experimental data points than others. So if these experimental data are a good reference, that means the predicted decoding times that are closer to them are better. And if you look at the different tRNA quantification approaches that were used to generate the tRNA abundances, then actually it turns out that counting gene copy numbers gives you the best model results that are closest to the experimental data points. So I should say that we don't know what the real decoding times are. It's possible that one of the experimental data sets is really accurate and everything else is inaccurate in relation to it. It's possible that our predictions are accurate and the experimental data points make mistakes, or most likely the real decoding times are somewhere in the center here, and the experimental data sets and our model data all make some mistake. That means they're differing from the real decoding times in yeast in vivo. But importantly, our best modeled decoding times are closer to some of the experimental data sets than the most different experimental data sets. So the difference here is smaller than the difference here, so we take that as a good sign. We like to see this as a sign that our models are producing data that are quite accurate. And So if we can accurately predict codon decoding times, like I said, then we can say a lot about ribosome dynamics, and we can design very efficient genes. So the range you have to play with, this is an exercise where we made a gene for firefly luciferase, and we made 3 variants. The worst variant we could come up with, the best variant we could come up with, and this is just wild-type firefly luciferase expressed in Baker's yeast. And you can see that we can really make expression levels measured by luciferase activity here, or by amount of protein in the Western blot here, that the expression levels go up a lot, about 25-fold compared to the worst variant. When you increase, you improve the decoding, you reduce the decoding times of the codon you use. You only use the fastest possible codons. And this effect comes from 2 things. So as you use the best possible codons, your RNA levels, the gray line here, your RNA levels become stabilized, and that's well known. So RNAs consisting of preferred codons are more stable than other RNAs. And of course, that in itself increases expression levels. But then over that, we also have gain of translational efficiency because the amount of protein increases more strongly than simply as a function of the RNA levels. So choosing optimal codons, as our models define, means that you stabilize the RNA and you improve translational efficiency, giving you higher protein expression levels. And this is just to say this works for other proteins as well. Slowly running out of time, so I'll accelerate a bit. And typically, so this is work we did with an industrial partner. Typically, our predictions sample 3 here, give you slightly higher yields than other optimization predictions do. If in general the protein you try and express doesn't cause strong problems. So we had one product here where the problem with expression is simply in the protein itself. It's not a problem of an inefficient coding sequence. And of course, if the protein is a problem, then making your coding sequence efficient doesn't really do anything. And this is the last result slide. So very recently, we have also been starting to apply these insights to making RNA vaccines. So if you remember the COVID pandemic, and who wouldn't, the COVID pandemic gave rise of the idea or gave us the first products where RNAs are injected into patients and the patient cells then decode the RNA and makes a protein in vivo. In the case of a vaccine, an antigen. This making proteins from an injected RNA has the same problems as making protein from a recombinant gene. You want to have an efficient RNA, and we can show that when we optimize RNAs using our decoding models, you can make vaccines that in mice, so these are mouse data, are more efficient. And so this is a comparison between our design 2, versus a vaccine designed to have the same sequence as the Pfizer vaccine, Comirnaty. And you can see that especially at high doses, we can achieve higher both B cell and higher T cell responses when we optimize our constructs. So in conclusion, we now understand a lot about the biochemistry of codon decoding, and we can use that to computational model how fast codons are being decoded. We can predict codon decoding times with good accuracy, provided we have good information on tRNA levels, and we do that in fungi, but actually in mammalian cells where tRNAs are transcriptionally regulated, that is still a real challenge, and we're much worse for mammalian cells than we are for fungi. And model-supported sequence design has applications in biotechnology and in medicine. And so a lot of this work on the vaccines was done with collaborators in Cambridge, but in general, we do a lot of our work with Mark's lab here at Kent, and the computational models were done mostly by my postdoc, Fabio Hedayolu, who doesn't like his photo taken, and so who isn't on this acknowledgement slide. And our work was funded by a number of people who we're very grateful to. So thank you very much for your attention, and if there are any questions and there is time, I would be very happy to answer them.